everyone. Thanks for Glider to have me here for this talk to share our modeling work on this free energy landscape, mainly about the protein local search on DNA. Exaggerate a bit here as if the protein wonders where to go. Okay, so in my lab, we model transcription machinery from microscopic to mesoscopic scale. Many focus on transcription factor search on DNA, RNA polymerase, mechanochemistry, and fidelity control, and a bit on DNA supercoiling regulation for transcription. So I'm going to focus on the transcription factor search on DNA, but now about global search, rather very focused on the part that when protein land on the DNA, search for the sequence locally. Okay, so this target search problem has been brought about in last century, 1970s, as people realized some of the transcription factor can locate the targets about 1 or 2 order of magnitude above diffusion limit. So the theory comes that suggests this facility diffusion model where the protein combines its 3D search with the 1D sliding, hopping, jumping, or transfer, intersegment transfer. So effectively, for the 1D diffusion part, people have measured diffusion coefficient effectively. So it's about this sub-nanometer stepping corresponding to the microsecond timescale. So for such a tiny spatial and this short time scale, it is actually very challenging for experiments to detect them. So we thought about that computation modeling may help to look particularly at the step-by-step of this transcription factor search on DNA. So previously, we used the all-atom molecular dynamic simulation to look at the small transcription factor move forward and backward on DNA for just one base pair. We also look at the during this process, there are like very delicate hydrogen bonds formed between the protein and DNA backbone all the time. And if you compare the configuration of the specific binding and non-specific binding, you will notice the change of the hydrogen pattern notably, and also the protein orientation will change notably. Okay, so I'm going to focus on this work published last year on Biophysical Journal, mainly on the free energy landscape using this minimum structure model. So in this model, we present the protein as a charged sphere with polarized charge distribution with positive charge facing the DNA, which is negative charged linearly. So we use this Coulomb interaction modulated by exponential, this dependence for this protein DNA electrostatic interaction. The range is about several nanometer, this length. And for specific interaction between protein and DNA, we imagine that there are, I mean, it's not just takes perspective, there are hydrogen bond contact between protein and DNA. So we model them by this Morse potential with directional dependence. Since we can not really evaluate the hydrogen bond energetically, so we actually use this position weight matrix measured bioinformatically to decide on particular sites. So that is C, T, A, G, these which base is preferred by this matrix. So with this specific interaction, it is actually sub-nanometer range. So correspondingly, we can see that non-specific interaction and the specific interaction then distinguished mainly by this hydrogen bond interaction. So for the motion, we use non-given equation to describe the protein translation on the DNA. And besides, we have to take into account the protein rotation, which is very important because a specific interaction is directionally dependent. So besides, we have to consider the helical path impact to the hydrodynamic of the system. Only in these take into account, we can generate this effective diffusion coefficients in 1D matching experiment measurements. Otherwise, it will be 1 to 2 order of magnitude higher without this helical path consideration. So with this treatment, we can get effective diffusion coefficients modulated by this ruggedness parameter. So basically, for different sequence segments, you'll see different kind of ruggedness. So now we have almost the full description of the system. Okay, so now we are ready to construct this 2D free energy landscape locally. So along this direction horizontally, this would be this along DNA direction, while the perpendicular direction here would be direction of effective dissociation. So for this 2D free energy landscape, we actually have two ways to map them to one dimension. One way is that we imagine that here at particular spots, we have this protein dissociates effectively. So there will be binding free energy defined at this particular spot. But then along DNA, we have different spots. Then we have delta G measures the relative binding free energy profiling. So this defines relative binding free energy profiling 1D. And the binding involves several nanometers. So this is a nanometer electrostatic zone that detects this profiling. So on the other hand, we can think about the protein make a very tight contact to the DNA and sliding on the DNA. So they even keep the majority of the hydrogen bond contact from time to time. So then we basically construct the diffusion, 1D diffusion profiling, take this small range, which is sub-nanometer of this movement per minute. Displays along DNA. So then we have these two effective 1D profiling. So you'll see that this diffusional one actually have more rocked shape, while the this 1D relative binding free energy will have more smooth shape, because for the binding thing, it has longer this length scale, so it does accommodate more degrees of freedom as a protein rotates. And we haven't modeled, but in some cases, protein can also have protein conformation fluctuation. So it will allow this difference more can become larger. So we basically can have this cartoon motions. Meanwhile, we can see that sometimes protein dissociates. So in that case, we'll have this this landscape in particular look at this dissociation. So if protein move along specific on DNA, so they have a chance to dissociate from time to time. But when they tighten the bonds, there is tiny chance they can also dissociate from the spot. But it's very small probability. On the other hand, they do have the chance to diffuse to the labor site for the diffusion for the dissociation. So if we compare these ABC paths, we see that the paths go to diffuse to the labor site or flaking site indeed has lowest barrier. So we suggest that the probability is actually largest if the protein want to dissociate from the specific binding sites, and which is largely dictated by the diffusion to the flaking site. So the take-home message here is we have this first, by definition of the profiling, we see this binding profiling accommodates more degrees of freedom. So it actually detects less amount of DNA sequence information compared to 1D diffusion landscape. But this 1D diffusion landscape will be pretty hard to measure because it really demands very high resolution, which is not achievable most of the time so far. From the measurement point of view, this 1D binding profiling is more ready to be measured. But the thing is, in most of cases, DNA segments involved in the measurements having flagging sequence. So beside this extra degrees of freedom, just around this dissociation distance, they also have this labor site, flagging site impact, further smear the information detection of this, if you want to measure the protein binding profiling by the way of protein dissociation. Okay, so to further illustrate this idea, we still use this minimal model, but two ways of measuring the binding free energy difference. So one way is we just pull protein away from DNA. So then we can measure work and get the free energy difference, or we can mimic experimentally as allow the protein to dissociate spontaneously among different sequences and measure the corresponding time and the rate. Okay, so the first way is as if we do further in MD simulation, like if you just pull the protein away from the center binding site, right? Then you get delta G between these two sites. And the second way actually corresponding to that this protein actually are able to dissociate from the central and the neighboring spot. So mathematically, you can prove that this corresponding this binding free energy will be smaller than the first way of measurement. Okay, so we really did the molecular dynamics simulation at all atom resolution to show that. So we first pull the protein away from DNA. So because it's not very slow, we use Jarzynski's method to average the exponential of this work distribution, get the delta G. Actually delta G first, and then we get a delta G between different sequences. But we worry about that if the protein rotation is not quite sampled enough, then we were using another way of sampling. We use this umbrella sampling to hold the protein at the different distance away from DNA with some constraints and reweight the process and get the free energy. So absolute free energy decreased a bit while the relative binding free energy still keep pretty much the same, in this case about 10 kBT. So then we compare this consistent simulation result with experiments. We see that the experimental measurement is about half. And then we change to another method that instead of pulling protein away from DNA, we change the protein sequence from one to the other, either with protein bound or without protein bound. So we can form this thermodynamic cycle, so still measure the binding free energy on these two spots. So with this method, we also have the computational measurement of this relative binding free energy, delta G, systematic larger than the experimental value. So indeed, if we inspect the experimental measurement in more detail, we see that it was the 15 base pair DNA segments all used, while this protein only have 6 base pair core binding sites, while the extra amount of sequences are flagging, so impact on the measurement. So we suggest it is this physical movement of the protein impact because this flagging sequence effects were just detected more from data science point of view, from bioinformatic data. So we kind of suggest a physical-based way of this movement of the protein interfere with this measurement. Okay, so summarize this part. We basically have protein able to diffuse 1D, while when it tries to dissociate from some notable binding sites, the majority of the time or the main path would be they go to the labor site, the flaking site first, and then dissociate. Only a very small chance they will dissociate on the spot. Okay, so a little bit more about this research is that we can really just uh, um, survey the stapling pattern of the protein on DNA. So think about that we do have the hydrogen bond formed between the protein residues and the DNA nucleotides schematically present here. So on a homogeneous sequence, so we can imagine, let's say, the hydrogen bond pattern initially. So they are forming sort of like a tiny eggs, legs, something. So in the beginning, they form like this configuration. If all of them move and recover the same configuration, we define them move for one base pair distance. Well, things won't be that easy to tell that on the inhomogeneous sequences or random sequences, because if they move for one base pair distance of whole movement, you may see the pattern doesn't recover, or maybe three base pair still doesn't recover until fourth base pair. In this case, they recover the pattern. So we see that this one stapling actually has included four base pair distance. So this we have more statistics on that. If we do homogeneous sequence, we can always locate this free energy minimum of diffusion profiling separated by one base pair of the labor energy minimum. So if we do this stapling time statistic, you will see it is exponentially distributed corresponding to single time scale. If we do that on the random sequences, there are still some one base pair stapes separated by some minimum. While in many other cases, we will see energy minimum of this diffusion separated by two base pair, three base pair, or four base pair. So we can get a survey of the histogram on how much this different size distributes. Then we also have the waiting time distribution. So then you will see that instead of exponential, they have power, more distribution indicates this miscellaneous time scale. So we do have this dedicated interesting prediction, but it is actually very challenging to validate experimentally because of the resolution of the measurements. So indeed, if we really want to test experimentally, likely will need to design some repetitive signature patterns on the DNA and do the simulation and to see experiments can detect some characteristic for the pattern. Okay, indeed, for these repeat sequences, people have found they are of significance, for example, in our human being and related to the disease pattern. So in a more refined study, people designed a repetitive sequence around the key binding motif and suggest the impact of this short pattern repeats on this binding site affinities. And this is quite irrelevant with our current working hypothesis that wouldn't have this homogeneous or pattern repeats. They make the diffusion pretty easy and also impact on the core sequence binding. So let me use the last 5 minutes to switch gear to more recent developments. So we combine physical-based method and experimental data for machine learning. So in order to see if we can predict the protein binding affinity as well as the flaking sequence effect. So we know that physics-based method, we can start with high resolution, but the computation become very expensive. And sometimes if we use more faster method, the accuracy becomes issue. On the other hand, we know experimentally there are different type of profiling data to allow this information of protein binding specificity to be learned, so that we indeed use some comparative fast but not very accurate physical method with the experimental data for this machine learning. Okay, the physical method we used is this MMGBSA method. So it's calculate the use molecular mechanics, the binding energetics between protein and DNA. And for the solvent contribution, however, they use the continuum model to calculate the solvent polar and nonpolar contributions. So that's if you have a molecular dynamics trajectory, you can, for each frame, calculate the protein DNA interaction energy, correction term, and solvent contribution. Traditionally, you add these terms together, assuming the linear superposition of them predict binding free energy. However, in current modeling, we only take those energetics without adding them linearly. So we assume that this machine learning model will take care of this presumably some nonlinear superposition of them. So the data we choose is genomic context, the protein binding microarray data. So it does have core sequences, flanking sequences, and with that, we can construct a balanced data set of hundreds sequences of DNA. So we have long bound, weakly bound, and strongly bound cases. So the protocol works like this. We run MD simulation for each system with protein at one type of DNA sequences, overall for about 200 sequences. And then for each system, we take this protein DNA binding energetics and solvation contribution all together and average through the trajectory and take them as input features. And we use a neural network or more traditional methods of random forest to support vector machine and this experimental data for regression or classification. So roughly the result will be if we just take this MGBA method without using machine learning versus experimental data, we'll see the correlation is pretty low. The classification is also not very good. But if we use a machine learning method for the regression, we improve notably the correlation, while for classification, we also improve notably. So prediction is just one thing we want to achieve. This framework, we want to do interpretation with these physical input features, so we basically can tell which are more important. But we realize that with current parameterization of this hydrogen bond, the prediction is not good because they, with this parameterization, they just do not find the importance of hydrogen bonding. And when we reparameterize by taking this prediction weight matrix, we definitely can improve further the regression and tell that hydrogen bonds do play important role as with other components like van der Waals, hydrophobic, to detect protein specificity on DNA. Then with this framework, we thought about to predict flanking sequence impact. For example, we take the same core sequence and one base pair, two base pair, three base pair kind of flanking sequences. We see the energetic do fluctuate more predicted with this more flanking sequence. But however, if we look at this particular core sequence with different flanking sequence highlighted in red, we see that prediction and the experimental data is not good at all. This is also consistent. So right now with our thinking that the protein basically has to move on the DNA to have this flagging sequence effect. So then it is something under working right now. So we want to manipulate the input features to see possibly we can improve the prediction on this flagging sequence impact. So I'm wrapping up this part, and thanks for my current NAB members and previous NAB members for the contribution to this work, and thank you for your attention.